Hello friends, welcome back to this video, and in this video we shall continue our study under genetics, but the section that we'll look at today is what we call as cell division. Now, a cell, we have already defined a cell in previous lessons to say it's a basic unit of life. They are like blocks that make up what? The entire organism. So, this cell that makes up an entire organism must be able to help us form new cells or also help us to form reproductive cells. And so, we must divide these cells. Now, on Earth, there is no such thing as a completely new cell. Why? Because every new cell will come from a cell that already existed. So that is why you usually have, when you have an offspring, the offspring will resemble the parent. Why? Because the genetic information may be the same or exactly the same as that of the parent. So as such, we define cell division as a process in which existing cells divide to form new cells. Now, this process of cell division can occur in sexual reproduction, which involves a male organism and a female organism. But it can also happen in asexual reproduction, where there's no need of two parents involved for the reproduction to take place. So as such, we have two types of this cell division. The first one, which we call as mitosis, and the second one, which we call as meiosis. Let us begin with the process of mitosis. Now, mitosis is a type of cell division, firstly, that occurs in asexual reproduction, which does not involve two parents for you to form an offspring. So this is part of asexual reproduction. Now, how does it work? So this type of cell division divides the chromosome number, or I would say this type of cell division would divide a cell into two. So let's say we had a parent cell like this. After undergoing this type of cell division, the cell will be divided into two like that. But then every cell in the human body, here we are referring to eukaryotic cells, particularly human beings, they have a total of 46 chromosomes. Now, in this type of division, which is mitosis, the cell number divides by 2, but those cells are called as diploid cells because those cells contain the original number of chromosomes they had before this cell division started taking place. So expect that this will have 46 chromosomes and this will also have 46 chromosomes like that. So that's the basic idea of this process of mitosis. It divides a cell into two daughter cells which are genetically identical. As you can see, they contain the same number of chromosomes. On the other hand, meiosis is a form of sexual reproduction. This one divides the cell into 4. So if you had an original cell like that, you have 1, 2, 3, 4. So it produces 4 daughter cells for meiosis, but these daughter cells are not genetically identical to the parent. They are very, very different because if the parent had 46 chromosomes, this one divides the chromosomes into 2. So this will have 23, 23, and 23. So these, because they are half the number of chromosomes, we term them as haploid. Haploid coming from the word half. Now, let's look at the steps that are involved for this process of mitosis and this process of meiosis to occur. So for a cell to fully divide, it must undergo some processes which we term as phases, right? Now, how do these phases follow up in the process of mitosis? So in mitosis, we only have 4 stages that are used for a cell to divide. Of course, we have a non-dividing stage, which is not in, part of the stage, but in that stage, a cell is just in its resting state. So we'll begin with interphase. So interphase is really not a phase. It's not a phase of cell division, but it's a phase that's included under mitosis, which shows that at that stage the cell is just resting. So under this stage, what are some of the key points you must remember for a cell that is dividing? So this is a cell, so I'll draw a cell with only showing its nucleus. So inside the nucleus, we will have what is called the nucleolus. And then aside from that, we have these thread-like structures, which we talked of being chromosomes. So what are some of the key points here when you are looking at this interphase stage? Firstly, in this interphase stage, it is called as the resting stage of a cell. Why? Because during this stage, a cell cannot divide or does not start to divide. And what is the reason for it resting or not dividing in this stage? The reason is simple. The reason is this. Chromosomes here appear to be a tangled mass of very long and thread-like structures. And like that, they cannot be viewed under a microscope, and so division cannot take place. So the basic reason why it's a resting stage is because these chromosomes have not condensed yet for division to start. Another notable point here is the nucleolus. In this phase, the nucleolus disappears. What else disappears then? What else disappears is this membrane that is surrounding this nucleus, which we call as the nuclear membrane. So this nuclear membrane disappears. Now, why does this nuclear membrane disappear? It must disappear to allow these chromosomes to be able to surround or go into the cytoplasm so that the cell division can now start to take place. So this cell division occurs where? It occurs inside the cytoplasm. So this nuclear envelope must disintegrate or break so that it can allow these things that are here to now go into the cytoplasm, and the division can occur in the next phase. Now, in this interphase stage, we also have what is called replication. So before a cell can even start to divide, it replicates. Why? Because remember, in a cell division, you must produce an offspring, but you must remain with your parent, your original one, isn't it? Yes. So a cell, or this chromosome here, will do what? It will replicate or produce one of its own kind again at this stage. So replication occurs at this interphase stage, and it is one of the longest stages in cell division, not in the stages of mitosis, because it's not a stage of mitosis, but in this process of cell division, it is one of the longest stages. So these are some of the key points I'll need you to master. Apart from that, you can also have centrioles. Centrioles move to opposite ends. So the centrioles will now move to opposite ends of the pods. Now, what are these centrioles? Centrioles are just structures that look like tubes. So those tubes are what form what is known as the spindle fibers, and we'll talk about it when you have started looking at their function later on when you're looking at the stages. But the basic function is to form what are called as spindle fibers. After all this has happened, the stage now, the process of mitosis now moves into the first stage, which we call as the prophase. Now, what are some of the notable key things here under the prophase? So in the prophase stage, what happens is that since in the interphase stage, the chromosomes appeared to be a tangled mass which were long and thin. But in this case, in the prophase, those chromosomes now start to condense. So as they condense, they appear to be thick and short. Then as they become very short, they begin to pair up. So we have this whole structure that looks like this here. This whole thing is what we call a chromosome. So this can be a chromosome, and also this, when you split it into two, it can also be a chromosome. But when a chromosome like this is attached to another chromosome at a point which we call the centromere, we now refer to as these two that have bonded together or attached to one another as chromatides. So they are only called chromatides when they are attached together to form this whole thing, which is a chromosome. When it is one like that, we call it as a chromosome. So that is why we say those chromosomes now condense into these structures, which will later join together at points called centromeres and form two pairs of what? Of chromatides. So this is a chromosome. So if we put two pairs of this, then, or if we put two of these, then it's a pair of chromosomes. So in this stage, that is where we have this, the condensation of the chromosomes. I think that's really the big thing here. Aside from that, we also have when they meet up and form a chromosome. Yeah, I think that's just the basic thing, the overall thing that happens here. Of course, the disintegration of the nuclear envelope also continues in this stage. But yeah, this is the most notable thing under prophase. Then the next stage now starts, which we call as metaphase. Now, in the metaphase stage here, meta means equator or going on the metaphase plate. Equator is the middle point of, if we are looking at the Earth, the middle or the center of the Earth. So in this case, metaphase refers to these chromosomes now, they will start to align up together on the metaphase plate, which we call as the equatorial plate or the equator. So in this stage, what divides this stage from these other stages is that these chromosomes now begin to pair up here on the equator. So you have this chromosome there. I hope it's visible. Then you have that chromosome. So you must keep in mind that here we just write two chromosomes because we cannot write 46 chromosomes, but we must have 46 chromosomes as for a normal cell in a human being. So here is what you have. You have these things that are called the spindle fibers there, and these are formed as a result of what? The centrioles that we talked of earlier. So you have these, which we call the spindle fibers, forming due to these tubes which are called the centrioles there. Now, this stage ends with these chromosomes just aligning on the equator. The next phase we have is anaphase. Now, in anaphase there, what you have is that these chromosomes will start to separate into individual chromatides there. So what happens is that they will now start to do what? to separate or pull away from one another. Now, what causes this pulling away from one another is these spindle fibers that are formed, and that creates the basic function of these spindle fibers. So it will look something like this, where you have this cell being, this chromosome being put there, and this one also being put there. So there will be a lot there. You can also have something like that. So they are now being put to what? To opposite poles of these spindle fibers or of the cell. And then the last stage that we have under mitosis is what we call as the telophase. In the telophase here, what are some of the notable things that happen? This separates or is different from the anaphase, because in telophase, this nuclear envelope now, or this nucleus, starts to break down so that it can form two cells which will have the same number of what? Chromosomes. Remember, this is mitosis, so we are trying to originalize or take back the number of chromosomes as we had initially. So the notable part here in this telophase is that firstly, the things that that disappeared during the prophase, like the nuclear envelope, the nucleolus, they will now start to form again. So telophase is just the opposite of what? Of the prophase stage. So in this stage, you expect that you have two cells that have formed, and we refer to this process usually as cytokinesis. So that's another process of mitosis. And why is this type of cell division important if they ask you? The first reason is for asexual production. The other reason is, I think overall it's just the asexual production, and also you can also talk of forming new cells in the body. For example, when a cell is damaged, for that cell to be repaired, it takes this process of mitosis. And apart from that, also forming of new cells in the body. So we'll look at meiosis in the next video. Thank you.